Hi, my name is Abby Robinson. I'm at 10th grade at Connor Weiser High School in Robezonia, PA, and my project dealt with the occurrence of antibiotic-resistant genes in the Schuylkill watershed. Antibiotic-resistant genes are genes that mutate to build up resistance to certain antibiotics. This is a huge problem in the medical field because these genes build up so much resistance that it makes some of our most commonly used antibiotics essentially useless. So, for my project, I decided to test multiple water sources from the Schuylkill watershed for these resistant genes. The ones that I tested for were sulfonamide, vancomycin, and methicillin. Methicillin is no longer used in the medical field because of built-up resistance, but resistance to it does contribute to the global problem of methicillin-resistant Staphylococcus aureus, MRSA, which kills about 20,000 people per year. Vancomycin and sulfonamide are both antibiotics that are commonly used to treat various bacterial infections, including the skin, bloodstream, etc. So, to test these water sources for antibiotic-resistant genes, I first gathered water from 5 different sources along the Schuylkill watershed. These included the Topahawken Creek, the east and west end, Blue Marsh, Maiden Creek, and the Wyomissing Creek. After this, I filtered out all of the isolates from the water using a 5 micrometer syringe filter. Then I extracted DNA using the Lucigen DNA extraction kit. After this, I put my water samples that I had just extracted and concentrated into a quantitative PCR. PCR works because it uses primers that have the same genetic sequencing as what you're looking for in your samples, and it attaches primers onto the end, and it keeps extending if you get the right results. So what happened was when I put my samples into the PCR with the same primers, I ended up with positive results, as you can see in the quad chart. The amplification curve is the curve right here. The baseline indicates 0 results, meaning negative, and that curve means that there were positive results. My results showed that 80% of the water samples I tested from were resistant to methicillin, 20% were resistant to sulfonamide, and 0 of them were resistant to vancomycin. Let's talk about these results. So 80% of them were resistant to methicillin, like I said, and that obviously contributes to MRSA, like I said earlier, which is a huge superbug and it's hard to cure since there's so much resistance buildup. As far as sulfonamide resistance goes, sulfonamide is used a lot in the medical field, so the fact that there was so much resistance built up in this river is concerning. 20% is a significant number, and I would like to run these tests again to see if that would keep going. I ran the PCR test twice. The problem with the antibiotic resistance in the river is that the resistance can reach the human microbiome through drinking water people drink out of the Schuylkill watershed. So if I were to do this experiment again to expand, I am trying to test for resistance to oxacillin and amoxicillin because their genetic sequencing is so similar to that of methicillin. So I'd like to run it again to see if the same genes were also resistant to oxacillin and amoxicillin, since they're more commonly used in the medical field. Overall, these results do pose a threat to human health. I'd like to run this test again with multiple more antibiotic-resistant genes and see what else I can find. Thank you!